We,
Sohnia,
and Jaanuni, are a couple of pals studying science in undergrad. We are not professionals.
though every episode is meticulously researched, mistakes do happen.
If you notice that anything,
And we mean anything.
we state is inaccurate, please let us know.
Your comments, suggestions, and queries are important in furthering our personal and audience's understanding of science.
Thanks for being a part of this discussion.
We appreciate you.
We really do.
Hey everyone,
Hey.
Happy New Year.
Wow, haven't seen you guys since last year.
And this is where I leave the call immediately.
Okay, goodbye. That's it.
Yeah.
Thanks for joining us. Welcome back to the first episode of 2022 by Beaker Bros.
Heck yeah.
It's almost
exciting.
a year since we started Beaker
Mhm.
Bros.
An exciting time. Look how far we've come. Number 25.
Ooh.
Is it 25 or? I think it's 25.
I mean, you wrote down 25, so I'm going to go with 25.
Right. Right. Okay. Well, Jaanuni, tell us a little bit what we're going to be talking about today.
Okay, for my folks in Canada, or even for my folks specifically in Ontario, we all know that with the holidays, there have been a surge of COVID cases recently. What's even more interesting is how we've been actually how the government has been giving out these free antigen COVID tests. So, before you want to see your pal before the holidays, or, you might have had a Christmas party or something, a lot of individuals are taking these tests beforehand to see if they have COVID or not, instead of getting a PCR test, because, correct me if I'm wrong, but you have to pay for the PCR test now.
Yeah, they changed that.
Yeah, so now these antigen tests are like at home kits that you can do like with yourself. But I think what I was really most interested in, like, from this was because I was like, oh, so you're saying this was an option the whole time. Why haven't we been like just doing COVID tests at home? But then I also came across that it's not that accurate. It's not as good as the PCR one. There are false positives, there are false negatives. And then there's so many different type of tests. So there's like the PCR one and then the antigen one, and it's a matter of which one's better, which one's worse. So this episode is essentially us kind of delving into the world of COVID tests and the different types, and yeah.
Out of curiosity, have you been able to get a hold of the at-home ones?
Not personally, but I've had friends who like were able to grab them. So like friends who had younger siblings that were given, like the school gave it to them, or friends that like working downtown Toronto apparently gives you like a better priority of like getting an abundance amount of tests. But I haven't. I know there's like pop-ups that people have lined up for, like they're at 9 a.m. and you would line up for God knows how long and get a test. But I have not.
Yeah.
You?
Yeah, I was just curious because, same thing for me. I know people that have gotten it from like schools, better access in certain areas, but I've not been able to get a hold of one.
Yeah.
So
there are
limited supply.
I know. They're like everyone wants one. It's the new thing.
It's the new trend.
It's a new trend. I know like in one of the at-home kits, at one of the at-home like boxes, there comes like 5 kits. So, to get one kit, you have 5 tests. Yeah. Okay. But we can start off with talking about the different types of COVID-19 testing.
Yeah, go for it.
Okay. So we know obviously that there are different ways that samples can be collected to test for COVID-19. So this can be through the nose swab or throat swab, an oral fluid swab, saliva sample, or a blood draw. But I know you were also mentioning how there's 3 main types of COVID tests. You're saying did you want to go ahead and take that?
So there's the nucleic acid-based test. So that's the PCR one that Jaanuni's going to dive into a little bit. Then there's the antigen one. That one was already said. And then the serological one, that's the blood test. And there's other types of serological antibody testing methods, but blood test is one of the most common.
Yeah. Just like a quick note was that serology tests aren't actually used to diagnose a current COVID-19 infection, since they don't detect the virus itself. But it means a positive like antibody test or a serology test means that you have the antibodies to the virus that causes COVID-19, but it can't tell you like how long ago you may have been infected or determine if you're protected from reinfection as a result of previous infection.
And in addition to that, so like the most common type of antibody that appears from when you get that sort of testing, if it's indicative of having COVID, is the immunoglobulin G subtype. But that one is like the most abundant antibody in your bloodstream. So it could literally appear for any sort of virus or like infection that you have.
Oh, okay.
So it's not like, even if you like have an abundance of that subtype, it's not necessarily that, oh, you have COVID. But if you have symptoms that align with COVID and you have that, that could be a pretty strong indication that, oh, maybe you do have COVID. Maybe you are positive.
It was like I was saying before, like you might have an increase in white blood cells.
Yeah.
It might mean you have cancer. It might mean something else, right? So yeah, it's, I want to say similar to that.
Good comparison.
Okay, so we did the serology one, but so the PCR one is also known as a nucleic acid-based testing, which is also called a molecular testing. So these actually test the virus's genetic material, aka the nucleic acids, and this is considered the gold standard, I guess, by Canada for diagnosing active COVID-19 infection in patients. Then there's the antigen one, which basically detects specific proteins on the surface of the virus. They're often called rapid tests, so like the rapid antigen tests people you might be hearing about. They're called rapid tests because they typically provide results in less than an hour. And antigen tests are actually designed to be used at a point of care by a healthcare professional or a trained operator. But there are lab-based ones, but I also think it's funny how we now have at-home kits. So technically the two main types are the antigen one and the PCR one.
Yeah.
If we want to go more into technically how they work, so with the PCR one, PCR, it's called PCR because of the polymerase chain reaction. So for PCR test, what you initially first do is get a sample from the patient. So this can be a nasal swab or a saliva one as well. Sorry, my voice. And so for PCR test, what you do after you get the sample is to amplify the genetic material. So even if there's a small amount of COVID genes in the patient sample, it can be detected, which I think is like super cool. And the amplification is actually done through a polymerase chain reaction, hence PCR, where once they take the sample, they treat it with an enzyme that converts the RNA into double-stranded DNA that we know. And then the DNA is mixed with a solution containing another enzyme called polymerase, and it's heated, which causes the DNA to separate into two single-stranded DNA pieces. And then the temperature is lowered, and polymerase, with the help of a small piece of guide DNA called a primer, will bind to this single-stranded
Primer.
DNA.
Sorry, you said it. The primer.
And then it will actually bind to the single-stranded DNA and make copies of it. So the primers actually ensure that only the COVID-19 DNA is being amplified and not the other DNA. And so now you have like two copies of the COVID-19 DNA from the original piece of your RNA. And then technically, not technically, this is how it works. The lab machines, they repeat these like heating and cooling cycles, like this whole amplification system at least 30 to 40 times, which means you're doubling the DNA until there's a billion copies of the original piece. And then the amplified sequence, so like the bunch of copies you have, contains a fluorescent dye that's read by the machine. And so this property of like the amplifying property of the PCR actually allows the test to successfully detect even the smallest amount of COVID-19 genetic material in a sample, which makes it like a really highly sensitive, is a way they used to describe it, and accurate test. There's even research just saying it has an accuracy of 100%. bold statement, but 100% indeed. So that's for the PCR test. It's a whole like you have a little bit, even if you have like the smallest amount, you got a bunch at the end, and that's how they're able to test because of the fluorescent dye and everything. For the antigen one, this one is, sorry, I just blinked out.
That's the surface or specific proteins, right?
Yes. Okay. So before we go on to talk about like the antigen testing, I think it's really important to mention what an antigen is. Antigens are substances that cause the body to produce an immune response, so they trigger the generation of antibodies. So these antigen tests are like they use lab-made antibodies to search for the antigens for the COVID-19 virus. So imagine like, you know, a bestie. You're trying to find your other bestie in the body.
Y'all at the club, you can't find each other.
Exactly. That's how the antigen test. And let's be honest, you can find your bestie in less than an hour.
You know what? Like, sometimes that's good timing.
That's great timing.
Yeah.
So just like you have the antibodies in your body, the ones on so there's a test strip. So the ones on the test strip will actually bind to any of the antigen in the sample. So if the antibodies bind to COVID-19 antigens, then a colored line appears on the test strip indicating the presence of COVID-19. Which is, I guess, I don't know if you guys have seen it, but it's like the little rectangular thing, like the at-home kits, how there's a little rectangular thing, and there's like a C and a T, and then there's like a little line. So the colored line is like, hey, your antigens and your antibodies, they're binding.
I was gonna say, kind of looks like a pregnancy test.
I was
You've
gonna
never seen those. Yeah.
Yes, that is also another way.
Wait, did you mention the reliability yet of them?
No, not yet. Did you want to go ahead and do that?
Like, I don't know as much as you, so this is me sort of just speaking out of my ass. But the main reason, I think, correct me if I'm wrong, that it's not as reliable is because like it doesn't do that amplification process, right? So that's why you have to wait that 5 days or whatever it is.
Yeah.
So it's just like detecting presence or whatever.
Yeah, so it is definitely less accurate than PCR because I think it's more those antigen tests are better for those who are symptomatic or who have a lot of virus in the system because again, like you don't have that amplification system, which means like even if there's like a small amount, you don't know if it's going to detect. I think again, back to like me, I was curious on how, because at first I was telling you earlier how I thought there were a lot more false positives that were appearing from these antigen tests, but I actually found out there's actually more false negatives. If we're talking about exact numbers, they're saying that, so like one of the popular antigen tests, they miss 15 out of 100 infections, so that's 15 false negatives, versus it gives you a false positive test about 1 in 100 times for like so individuals who aren't infected.
Real quick, do you want to explain the difference between false positive and false negative?
Yes, so one is a false negative, which means you have COVID, but it doesn't tell you that you have COVID. And then false positive is when it says you have COVID, but you don't have COVID. I think that's the best way I can explain it.
Yeah, that's exact. One thing from what you just said that I think is pretty interesting. So like in stats, we learn like there's something like it's called statistical significance. And normally if the p-value is less than 0.05, so that means like there's a less than 5% chance of it being like not the outcome that you have.
Yeah.
Then like that's considered like significant and then it's used or whatever. But you're saying 15 out of 100 are inaccurate. So technically that's not statistically significant in terms of its ability to like detect, but it could be clinically significant. So like there's a difference between the two. So if it still has like some degree of effectiveness, as it clearly does, like it's still getting 85%, even though it might not be like according to standards of like statistical significance that we see in the lab, it's still very much clinically significant, and that's why it's like, used in the way it still is.
Yeah. I also think with that, it's like the timing of when you take the test, because like you were saying earlier, like if you take the test in the early stages of having COVID-19, you're not going to have a lot of the viruses or not a lot of the antibodies. So this test won't be able to detect for that. That's why if you're not like, this is what mind boggles me. Is just the fact that like they're saying for those who are asymptomatic to take these rapid tests at home, even though these tests are not the best for testing if you're asymptomatic. But I know you were saying, what was it? You have to wait 5 days or something before you should take the test.
Yeah, like that's what the new like incubation period guidelines are in Ontario. It might be, or in Canada, it might be different elsewhere, but that's what it is here.
Yeah. So like I guess you can wait. I think that's why they tell you to wait a couple of days before taking one, because at that time they hope it's a bit more accurate than it was on day 1 versus day 5.
Yeah.
But that's it for what they are and how they work. We can sorry, what was I gonna say? Sonia, have you ever had a COVID test done?
I have had a COVID test. I've had a couple. I had one at school. They what did they do? Yeah, they put it up the nose. And then I had another one that was at the, what's the name of the hospital? I don't have to say the name of the hospital, but I had one done at a hospital just as like a precautionary check that I had to get done. But you've had like dozens.
I've more than like
hundreds.
A crazy amount. I was basically working at a long-term care home, and at that time, the government of Ontario, before like vaccines were like officially it was, I think, until there's like 80% of the population that was vaccinated, so I had to get tested literally every week, and it was the one that was up to the nose, not the through the swab, none of that stuff. So it was a great experience.
Sure.
I've done more than 50 COVID tests. But hey, it's fine. I've done.
It makes you stronger. Pardon?
I've done like COVID tests for other people, like the up nose one. It's an interesting experience. I think we were mentioning in this earlier how we're like, why does it go up to the nose, right? Like we've seen there's so many different types.
Great segue.
Why is the nose considered the gold standard and stuff? So I think what I've come across, because Sonia and I were actually having a hard time trying to figure out why the nose, like why up the nose. And I think the best way, one of them was explaining how essentially that they need to collect a sample from the entire passageway, so the back of the throat and at the end of the nasal passageway. So again, you can either do that through the throat or through your nose. And if you do it through the throat though, there's the gag reflex. So I think, as a precautionary thing, they do it up the nose. And they're able to do it like that. Yes, it's uncomfortable, but you might not yak from that.
Yeah. But in saying that, though, there's also been studies that we came across. Like there was one published earlier this year in Nature that says like collecting samples through saliva. So whether it be just like, I don't know, like swabbing your cheek or, I don't know, spitting in a test tube. I don't know how else they would do it. Yeah. But like that's also like effective as well. In like there's a similar level of effectiveness. Like it's statistically significant.
Yeah.
In terms of the similarity.
Sorry, I totally think that. But yes, it's kind of cool to see how like it's been evolving too. Like at one moment they're saying like this is better, but then there's research that's saying that this is better. So it's really interesting to see how like science is continuously adapting to new research.
Yeah, and it's crazy how fast it does it as well.
Yeah.
Like there's one day there's a study coming out of like Germany that says, oh, this is like the most effective thing. The next day it's like, or 2 days from then that moment it's published, then it's like global guidelines are changing. It's crazy.
Crazy how the world works.
Yeah.
But did you want to add anything else, Sonia?
I think we did a good job covering it. I'm trying to think. I don't think we did the different types of collecting samples. So like we didn't say it explicitly, but we kind of explained it.
Yeah.
yeah, I think that's it for the topic.
Yeah. Pretty interesting. You never know. Maybe in a couple of months, there's a different way to test for COVID. I
Yeah.
wouldn't be surprised. Hopefully, again, it's not up the nose. The ones the at-home ones, though, is what I've been hearing is it doesn't go all the way back. It's like
Yeah.
1.5 inches up your like nasal passageway.
Yeah.
That was interesting.
Mm-hmm.
Um,
Yeah, the actual like swab itself is smaller. It doesn't like
yeah,
go that far.
like
Yeah.
go to the brain, basically. Which I thought was interesting because I was like, I literally thought they were doing the ones that like basically you're kind of scratching your brain in a sense. It's
No,
not.
like for a little bit I thought that as well. Like that's
No,
why like
bold of them to
literally like the swab disappears up your nose. It's like I guess it's in my brain now.
I'm pretty sure as a kid we were told not to put things up our nose, not to put anything in our ears, but here they're like,
Yeah.
yeah, go ahead.
Pop, right up the nose.
With that being said though, I think that's it for this week's episode.
Yeah.
Stay safe, my dudes.
Yeah.
Get tested
All right.
if you wanted to.
If you can,
If you
and
can.
if you need to, and if it's necessary.
Yep.
But yeah. All right.